What's up guys, good evening. This is your NSEJS Biology Master Teacher and welcome back to the session number 3 for NSEJS 2025 Biology Basic to Advanced series. I hope each one of you would have watched the last two sessions in which we had started this very important and very difficult unit, that is biomolecules. So in the very first session I had taught you about what are biomolecules, what is cellular pool, how to do the chemical analysis of cellular pool, what are metabolites, primary metabolites, secondary metabolites, what are carbohydrates, the classification of carbohydrates, what are monosaccharides, oligosaccharides, polysaccharides. Then in the previous session I had taught you what are amino acids, the classification of amino acids, and finally the organization of the proteins, right? The primary structure, secondary structure, tertiary structure, and quaternary structure for proteins. In this session I will be continuing with the classification of the proteins. So again, the classification of proteins, it is one of the most important topics that we have in this particular chapter. So without any further delay, let's start with the session. I'll take you to that particular page. Yes. So today we'll start with the classification of proteins. So the proteins, they are classified on various parameters. The very first parameter is the classification of proteins on the basis of their isoelectric point. So isoelectric point is something that you read in chemistry. I will give you a brief about what an isoelectric point is. So first of all, let me ask you this question. What are amphoteric compounds? So if a compound can react with an acid as well as a base, it is considered as amphoteric. So the amino acids, they are also amphoteric. Yes. So an amino acid can react with an acid as well as a base. So amino acids are considered as to be amphoteric. So I'll write this point that amino acids are amphoteric. Now, since they are amphoteric, so they can react with acids as well as bases. Since proteins are, again, they are the polymers of amino acids, so proteins are also amphoteric in nature. Proteins are also amphoteric in nature. Now let me draw the basic structure for an amino acid, the general structure for an amino acid. So in the last class I had taught you that amino acids are alpha amino substituted carboxylic acids, or they are substituted methanes, right? So again I will draw the structure, the general structure for an amino acid. So it will look something like this. Okay, so guys, when we look at the two functional groups, NH2, the amino group, and the carboxylic acid group, so these groups are ionizable, right? And the ionization will be different at different pHs. Okay, so let me draw the relevant structures for you. Let's say when you are at a very acidic pH, so how the ionization will look like. So in that scenario, the amino group will get protonated. So overall, you will have a positive charge. So this is going to be like the cationic form of the amino acid at a very low pH. At a very low pH, when you have a higher concentration of the H plus ions, the whole amino acid will get protonated. Now then, when you have the basic environment, when you have the basic environment, how the amino acid would look like. So this is how the amino acid will have overall the negative charge. Okay, so it will be in its anionic form. So the ionization of the cationic group and the anionic group depends on the pH. Now there will be a pH when both the groups will get ionized and overall you will get a zero charge. Let me just draw that particular stage for you. So if you can see, at this particular stage that I have drawn in front of you, the overall charge over the molecule is 0. The net charge is 0. So this is known as the Zwitter ion. This is known as the Zwitter ion. And the pH at which this happens, it is called as the isoelectric pH or isoelectric point. So isoelectric electric pH or isoelectric point, right? So the pH at which the amino acid overall it has the overall zero charge or no charge, that pH is called as isoelectric point. That pH is called as isoelectric point. So now this particular thing I have told you about the amino acids, but what about the proteins? So proteins are again amphoteric in nature, they are also amphoteric in nature. So at the physiological pH, now what's the physiological pH? That is 7.4. so at the pH 7.4 we will look at the overall charge of the protein, right? So I'm talking about the physiological pH, that is 7.4. So at that particular pH, a protein can have a overall positive charge, it can have an overall negative charge, or it can have no charge, or it can be neutral. When a protein has overall positive charge, it is called as the basic amino acid, basic protein. It is called as basic protein. When a protein has an overall negative charge over its surface at the physiological pH, it is called as acidic protein. And when it has no charge, it will be called as the neutral protein. So basic proteins. So basic proteins generally they tend to have more amount of basic amino acids like arginine and lysine. So the basic proteins they are rich in amino acids like lysine and arginine, which are basic amino acids. We can take an example as well. So for the basic protein, you have the example of histone proteins, right? So they are basic proteins only. Overall, they have a positive charge as the physiological pH. Then acidic protein. So they will be rich in aspartic acid and glutamic acids. They will be rich in aspartic acid and glutamic acid. That means the acidic amino acids. Glutamic acid, right? So example mean the blood proteins. Okay, the blood proteins. Most of the blood proteins are acidic proteins actually. The example include blood proteins. Then neutral proteins. So overall they will have the net charge will be zero. So basically it will have the most of the amino acids that will have their isoelectric point somewhere around 7.4, right? So I will just write the examples for the neutral proteins such as hemoglobin. Okay, so this is the first classification of the proteins that is on the basis of their isoelectric point. First of all, I taught you what an isoelectric point is. It is the pH at which the amino acid has overall zero charge, right? So now the proteins, they can either be a basic protein, an acidic protein, or a neutral protein. Basic protein, when you have the overall positive charge, basic proteins are generally rich in basic amino acids such as lysine and arginine. Example includes histone proteins. Then we have the acidic proteins at the physiological pH of 7.4. Overall, they have a net negative charge. Example, they are rich in acidic amino acids such as aspartic acid or glutamic acid. Example, most of the blood proteins. Then we have the neutral proteins. So the neutral proteins overall at the physiological pH 7.4, they have no charge or overall zero charge. Example include hemoglobin. Now let's move on to the next category, that is on the basis of the shape. So on the basis of the shape, proteins can be classified as fibrous protein or globular protein. You can have fibrous protein or globular protein. So I'll just write it over here. Proteins on the basis of the shape. The proteins on the basis of their shape, they can be fibrous protein or globular protein. Fibrous proteins or globular protein. So now the fibrous protein, as the name suggests, they will look something like fibers, right? So they are thread-like. Okay, so they are thread-like polypeptide chains. They are thread-like. They are tough. They are tough, non-enzymatic, non-enzymatic structural proteins. Structural proteins. So the fibrous proteins, they are thread-like, tough, non-enzymatic structural proteins, whereas the globular proteins, they are rounded, enzymatic or non-enzymatic proteins. So the globular proteins, their shape, so they are rounded in their outline, rounded in outline, rounded in outline. They can serve structural or they can serve enzymatic or non-enzymatic purposes. They can be either enzymatic or non-enzymatic in nature, right? So now let's talk about the structure. So the fibrous proteins, they generally tend to have a secondary structure, whereas the globular proteins, they tend to have the tertiary or quaternary structure, right? So for the fibrous protein, let's take the example of keratin. So keratin is a fibrous protein. So keratin basically exists in its secondary structure only. So generally have secondary structure, whereas the globular proteins, they have tertiary or quaternary structure. Generally have tertiary or quaternary structure. Okay. Now we can move to the next point, that is the fibrous proteins. They are completely insoluble in water. Globular proteins, so the smaller globular proteins, they are soluble in water, but as the size increases, the water solubility decreases. Okay, so I'll just write the same thing over here, that these are insoluble in water. For the globular protein, so the globular proteins, let's say they are the smaller globular proteins, they are soluble in water, but the water solubility decreases with the increase in size. Okay, so smaller globular protein, they are soluble in water soluble, and then I will write that water solubility decreases with increase in size. Okay, so now we can look at the examples for the fibrous protein. So for the fibrous protein, the example includes keratin. Now keratin serves structural purposes. You can have myosin, it is a contractile protein, so they can have structural roles, or some fibrous proteins are some structural proteins have contractibility as well, whereas in globular proteins, they are never contractile. They can't have contractibility. They can have the enzymatic role, or they can have some non-enzymatic roles. Okay, so let's talk about the example of the globular protein. Okay, so here I have written keratin, and the next one is myosin. Okay, elastin. Correct. Then for the globular protein, let's say albumin, and the serum globulins. So here you have the examples, right? So I have taught you about the classification of the proteins on the basis of their shape. Correct? Easy, easy peasy. I hope everything is clear till here. Let's move on to the next criteria, that is on the basis of their function. So this one is going to be quite lengthy. I'll make a flow chart for you so that you can have the basic understanding, right? So again, we are talking about proteins. So proteins now. Proteins, on the basis of their function, they can have, like, they can be enzymatic or they can be non-enzymatic. The first one is going to be this thing, right? So they can be either enzymatic or they can be non-enzymatic. Let's cite some example for enzymatic proteins. So you have DNA polymerase, you would have heard about amylase, you would have heard about trypsin, etc. So there are ample of enzymatic proteins. Now, if a protein is non-enzymatic, that what can be its possible function? Let's talk about this. So if a protein is non-enzymatic, what functions can it perform? Okay, so let's write that. Yes. So first of all, it can have the structural purposes. It can serve structural roles, right? So it can be a structural protein. Next, the storage proteins. You have the storage proteins. Next, transport. Hormonal. Then you have the protective or defensive. So we can have the protective proteins. So when example of structural protein, let's write keratin. For storage, we can write these are the examples I'll mention. These are the examples. Keratin, storage mein ferritin, let's write ferritin. So ferritin is like the storage protein for iron, ferritin. For transport, the most popular one, hemoglobin. For hormonal, you can have insulin, glucagon, etc. Insulin, glucagon, growth hormone. Growth hormone is also proteinaceous in nature. Hormone. Growth hormone is also called as somatotropin. So when we'll do the endocrine chapter, I'll tell you about the classification of the hormones and then I'll give you more examples about the hormonal proteins. And the protective one, so here you have the antibodies. You have antibodies, you have complement proteins, you have fibrinogen. There are ample of examples again. But I'll give you the most important one. Right now, if you'll refer to some international publications, then you can find more categories. Right, you have receptor proteins. When it comes to receptor proteins, they are GPCRs. Okay, G protein coupled receptors. Then you have the toxin proteins, etc., etc. So you will find more categories. But I hope up to the level of NSEJS, this much classification is enough for you. Now let's move on to the next one, that is on the basis of the constitution. So on the basis of the constitution, like the proteins, the proteins can be classified into three major categories. So proteins, first, you can have the simple proteins, you can have the conjugated proteins, or you can have the derived proteins. You can have simple proteins, you can have conjugated proteins, or you can have the derived proteins. Derived proteins. Now let's talk about what are simple proteins. So the simple proteins, they will be made up of amino acids only. Example में histone. So made up of amino acids only. Amino acid short में it is AA, made up of amino acids only. Example includes histones, keratin. Now the next one, that is the conjugated proteins. So conjugated proteins, they will also have some non-amino acid prosthetic groups. What they will have? Non-amino prosthetic groups. Non amino prosthetic groups. Now we can talk about some examples, right? The nucleoproteins. There are chromoproteins, metalloproteins, mucoproteins. Okay, you have glycoproteins, etc. So the example include nucleoproteins, chromoproteins. So chromoprotein, it's a category. You can take an example of a chromoprotein like hemoglobin. So hemoglobin mein, the non-amino prosthetic group is going to be the heme group, which is made up of, which is an iron porphyrin complex. So chromoprotein. Then the next you can have metalloproteins. Metalloproteins mein again one example I gave you, that is the ferritin. So it has iron. So ferritin, metalloproteins. Then you have glycoproteins. They are the component of plasma membrane. Then you have the mucoproteins. Etc. Right? So again, this much I believe is enough. Now derived proteins. So they are derived from the proteins as the name suggests. So they are derived, we get them from the proteins through either denaturation or coagulation or breakdown. So I'll write obtained through. What the terms did I mention? Denaturation, coagulation and breakdown. Denaturation, when the proteins get denaturated or coagulated or get digested, broken down, then you get derived proteins obtained through denaturation, then coagulation and breakdown. Can you guess some examples? So in the digestion chapter, if you would have read it, so you find the term proteases. So proteases, they are derived proteins. Fibrin is a derived protein. So example include proteases, fibrin, etc. Right? So we can again do a quick revision of this. So proteins, on the basis of their constitution, they can either be simple protein that is made up of amino acids only, example in histone and keratin. You can have the conjugated proteins, that means they are going to have a non-amino prosthetic group, example includes nucleoprotein, chromoproteins, metalloproteins, then we have the glycoproteins, mucoproteins, and then we have the derived proteins. So they are when the proteins undergo denaturation, coagulation or breakdown, we get the derived proteins. Example include proteases, fibrin, etc. So this is the next parameter, that was on the basis of classification, on the basis of constitution. Now on the basis of quality. So on the basis of quality, proteins can be classified as complete protein or first class protein or incomplete protein or second class protein. Okay, so on the basis of quality, the proteins, they can be classified as complete proteins, or you can have incomplete proteins. Now the complete protein, so they are called as first class protein. And the incomplete protein, that is the second class protein. Okay, so you would have heard about, like, I hope you would have watched the ad about the milk additives, right? The Horlicks or Bonvita Complan, these brands, right? So they offer complete proteins. So what are complete proteins? Complete proteins are basically the proteins that have all the essential amino acids. Whereas incomplete proteins, so these are the proteins which lack any of the essential amino acids, right? So complete proteins, that means they are going to have all the 9 essential amino acids. So that's why they are called as first class protein. So the proteins from the animal sources, right? Meat, chicken, fish, egg. Okay, so the proteins that you're really getting from here, they are going to be the first class protein or complete proteins. Whey protein is a complete protein. But the protein that we get from plant sources, right? The plant proteins, I should say. So they are the incomplete proteins. Okay, so that's why doctors advise that we should add dairy products. Why? Because dairy products, they are derived from the animals and they have the complete proteins. So now, as I said, the essential amino acids. So you should know what are the essential amino acids. So the essential amino acids, I have added this table for you. Essential amino acids, so these amino acids cannot be synthesized in our bodies and we need to obtain them through our diet, right? So the example include histidine, isoleucine, leucine, lysine, methionine, phenyl, alanine, and threonine, and tryptophan, valine. So these are the essential amino acids. Okay, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. So these 9 are the 9 essential amino acids. Then we have the non-essential amino acids. So the non-essential amino acids, these are the amino acids which we which get synthesized in our body only. We have the genes that are responsible for the synthesis of these amino acids, right? So alanine, asparagine, aspartic acid, glutamic acid, hydroxyproline, taurine, theanine. So these are some non-essential amino acids. And then we have the conditionally essential amino acids. What are conditionally essential amino acids? So when your body is under stress, right, these under stress, let's say some illness or some other issues, right? So your body starts to get deficient for these particular amino acids, and in that scenario these amino acids become conditionally important. So arginine, cysteine, glutamine, tyrosine, glycine, ornithine, proline, serine, these are essential, these are conditionally essential amino acids. Matlab, in normal condition your body will be synthesizing them in the desired quantities, right, in the optimum quantities, but when we are under stress, okay, so the level of these amino acids, the levels get reduced, and in that scenario you will have to obtain them through your diet. So the essential amino acid, non-essential amino acids, and the conditional essential amino acids. Okay, so here I'm done with the classification of the proteins. I'll give you a brief revision about it. So the proteins classification first is on the basis of the isoelectric point. What is the isoelectric point? The pH at which the overall charge over the amino acid is 0, right? So physiological pH of 7.4, some proteins can have overall positive charge, then it will be called as a basic amino or basic protein. Basic proteins are rich in basic amino acids such as lysine, arginine. Then we have acidic protein, they have an overall negative charge at the physiological pH. They are rich in acidic amino acids such as aspartic acid, glutamic acid. Example include blood proteins. And we have the neutral proteins. So the neutral protein overall they have the 0 charge or no charge. So the hemoglobin. Next is on the basis of shape, so fibrous protein or globular protein. When I say fibrous protein, so they are thread-like of non-enzymatic structural proteins. They generally have secondary structure. Example, they are water insoluble. Example include keratin, myosin, elastin. Then we have the globular proteins. They are rounded at outline. They are enzymatic or non-enzymatic. They generally tend to have tertiary or quaternary structure. Smaller globular proteins are water soluble, but the solubility decreases with the increase in the size. Example includes albumin, serum globulins, etc., etc. Then we have the next category that is on the basis of the function. So proteins can be enzymatic or non-enzymatic. Enzymatic one may example include 3Na polymerase, amylase, trypsin, etc. Non-enzymatic one, so structural may keratin, storage ferritin, transport hemoglobin, hormonal, insulin, glucagon, growth hormone, protective. So antibodies, there are complement proteins, fibrinogen, etc., etc. Next, on the basis of the constitution, proteins can be simple proteins, conjugated proteins, or derived proteins. Simple proteins, so made up of amino acids only, histones, keratin. Then you have the conjugated proteins, so they have non-amino prosthetic group. nucleoprotein, chromoprotein, metalloprotein, glycoprotein, mucoproteins, right? And then you have the derived proteins. So these proteins are obtained through denaturation, coagulation, or breakdown, proteosis, or fibrin, etc. Now, on the basis of quality, complete proteins and incomplete proteins. Complete proteins will have all the 9 essential amino acids. Incomplete proteins, they like the plant proteins, they will lack in one or more than one essential amino acids. What are the essential amino acids? The amino acids which cannot be synthesized in our body, we need to obtain them through our diet. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. Okay, then the non-essential amino acid, we have genes that the genes can synthesize these amino acids, right? But of the genes, they give rise to the pathways through which these amino acids can be obtained. Alanine, asparagine, aspartic acid, glutamic acid, hydroxyproline, taurine, theanine. And then we have the conditionally essential amino acids. When in the condition of stress, the level of these amino acid decreases in our body, and in that scenario we need to obtain these through our diet: arginine, cysteine, glutamine, tyrosine, glycine, ornithine, proline, serine. These are the conditionally essential amino acids. Now we can start with the next topic, that is nucleic acids. So the nucleic acids, the genetic material, right, the DNA or the RNA. So I'll write the nucleic acids, the nucleic acids, the monomeric unit, right? So the building blocks are nucleotides. So first of all, I'll teach you about what the nucleotides are, then we'll talk about what the nucleic acids are. So nucleic acid, that is the DNA or RNA. So the nucleic acids, the building blocks So the building blocks are called as nucleotides. Now nucleotides. So what is a nucleotide? So nucleotide, you can say it is a condensation product of 3 chemicals. Nucleotide is a condensation product of 3 chemicals, right? So what these 3 chemicals are: first you have the beta pentose sugar, the beta form, right? So in the very first session I had taught you about the beta sugars. So beta sugar, right? The next is the phosphoric acid, right? And then you have the nitrogen bases, the nitrogenous bases. Okay, so so the nucleic acids, DNA or RNA, they are the genetic material, right? So building blocks of the nucleic acids are the nucleotides. What is a nucleotide? So it is the condensation product of 3 chemicals. The 3 chemicals are the pentose sugar, phosphoric acid, and the nitrogen bases or nitrogenous bases, right? So now we can start with the structures first. So for the pentose sugar, you can either have the ribose or deoxyribose, right? So you can have ribose or deoxyribose. Now, ribose in the case of RNA, deoxyribose in the case of DNA. Ribose, the formula for ribose is C5H10O5, right? So CN, H2 and ON, the general formula for the monocyclic is deoxyribose. So at carbon number 2 of ribose, deoxygenation happens, and that's why there is a loss of oxygen, and you are left with ribose with 1 oxygen at carbon number 2 lost, right? So the formula will become C5H10O4. So this is the formula, ribose, deoxyribose. Then comes the phosphoric acid chemistry, H3PO4, right? H3PO4, if I have to draw the structure for phosphoric acid, so it will look something like this. right? This is the structure for phosphoric acid. I have already taught you about the structure of ribose and deoxyribose. That's why I haven't drawn it. Phosphoric acid, the structure is given over here, and then comes the nitrogen bases or nitrogenous bases. So what are nitrogenous bases? So these are heterocyclic compounds. These are heterocyclic compounds. Okay, let's talk about the further classification of the nitrogen bases or nitrogenous bases. Okay, so nitrogenous bases, you can have the substituted purines or you can have substituted pyrimidines. Substituted purines, substituted purines, or you can have substituted pyrimidines. Substituted pyrimidines. Okay, so you can have substituted purines or you can have substituted pyrimidines. What is a purine? So purine, if you look at the structure, purine, like you have the pyrimidine ring fused with an imidazole ring. So purine, pyrimidine ring fused with imidazole ring. Okay, so imidazole ring fused, pyrimidine ring fused with the imidazole ring will be the purine. So now what I have taught you, I'll just give you a quick recap. We have the nitrogen bases. The nitrogenous bases, they are further classified as substituted purines or substituted pyrimidines. Purines can all, they, what are purines? So purines are basically the pyrimidine ring fused with the imidazole ring. Okay, so I'll draw the structure for the pyrimidine ring first. Okay, so if you can see, heterocyclic, that means in the ring you will have atoms other than carbon. So here you have nitrogen, and then I'll just complete this. I'll draw the double bonds now. So this is the pyrimidine ring. Now substituted pyrimidine, that means you will have the substitution over the ring. Okay, so this is the pyrimidine ring. Then I'll draw for the purine ring. Okay, so purine means pyrimidine ring. As I said, pyrimidine ring fused with the imidazole ring. So first of all, I'll draw the pyrimidine ring. Okay, so this is a purine. So pyrimidine ring and the pentagon ring that you can see, that is the imidazole ring. Okay, so pyrimidine ring fused with the imidazole ring at 4 and 5th carbon is the purine. Okay, so numbering, you need to understand the numbering. So in your syllabus, the numbering for the heterocyclic compound is not a part of your syllabus, right? So you don't need to know how to, what are the rules behind this numbering that I'm going to show you. You just need to remember the numbering. Okay, so for the heterocyclic compound, the rules are different from that you have read for, like the general rules that you have read from the IUPAC. So here the rules are little different, but the numbering will look something like this. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have nitrogen atom at 1, 3, 7, 9 position. Okay, this is what you need to remember. Nitrogen atom at 1, 3, 7, 9 position. Okay, so anti-clockwise over the pyrimidine ring for the case of purine, and here the how the numbering will look like. So the numbering is done in this manner: 1, 2, 3, 4, 5, 6. Right? 1, 2, 3, 4, 5, 6. So this is the numbering for the pyrimidine. Now I'll show you how have I done the numbering. So this is what you need to remember. Okay, so right now I have just shown you the basic structures for purines and pyrimides. I haven't drawn it for the substituted purines or the substituted pyrimides. First of all, let's move on with the further classification. So the purines, you have the adenine or guanine, right? For the purines, you have adenine or guanine. Okay, and for in the case of the pyrimidine, substituted pyrimides, you have three. You have three. Okay, so the first one is cytosine, thymine, and uracil. Okay, so here you have it. Okay, now let's talk about so adenine, guanine, cytosine. These three are common in both DNA and RNA. Okay, so these three are both in DNA and RNA, present in both DNA and RNA. Thymine only occurs in your DNA, uracil only occurs in RNA. Okay, so DNA will have adenine, guanine, cytosine, and thymine, whereas RNA will have adenine, guanine, cytosine, and uracil. Okay, it will not have thymine. In the place of thymine, it will have uracil. Now, again, we'll talk about the structures. So, I'll tell you the chemical name for these compounds. Okay, it will make you memorize these structures in a easier manner. Okay, so let's say like adenine. So adenine is also known as 6-aminopurine. It is also known as 6-aminopurine. It is known as 6-aminopurine. Guanine is known as 2-oxy 6-aminopurine. It is known as guanine. So guanine is known as 2-amino 6-oxypurine. 2-amino 6-oxypurine. Now, so the cytosine is known as 2-oxy 4-amino pyrimidine. Cytosine is known as 2-oxy 4-amino pyrimidine. Thymine is known as 5-methyl uracil. 5-methyl uracil. Uracil is your 2,4-dioxy pyrimidine. 2,4-dioxy pyrimidine. Okay, so I'll tell you how you can memorize this, right? So adenine, so it is 6-aminopurine, guanine 2-amino 6-oxypurine. For cytosine, it is 2-oxy. Now there you had like 2-amino. Here you have 2-oxy. 2-amino in case of guanine, right? 2-amino 6-oxypurine. Here you have 2-oxy 4-amino pyrimidine. Thymine में 5-methyl uracil, very easy. Uracil 2,4-dioxy pyrimidine. Now when I know these names for 6-aminopurine, 2-amino 6-oxypurine, cytosine, 2-oxy 4-amino pyrimidine, 5-methyl uracil, 2,4-dioxy pyrimidine, then it becomes easier for me to draw the structures. Now I'll teach you how you can draw the structure. So let's start with the very first structure, that is of the adenine. Adenine. Now, as I taught you that adenine is also known as 6-aminopurine. It is also known as 6-aminopurine, right? So, first of all, let's draw the basic purine ring. Let's draw the purine ring first, right? So, Okay, so this is the basic purine ring. I haven't drawn the double bonds now. First of all, what I'll do, I'll read the name 6 amino purine. Okay, so remember the numbering. How had I done it? 1, 2, 3, 4, 5, 6, 7, 8, 9. This manner. Okay, so 6 amino, that is over the 6th position. I will put the amino group. So I'll erase this. I'll put amino group, that is NH2 over here. Okay, this is the 6th position, and then I'll draw the double bonds. Okay, so here, 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 here. This is the structure for adenine. So easy, right? Now let's draw it for guanine. Guanine. So guanine, I told you 2 amino 6 oxy purine. 2 amino 6 oxy purine. Okay. So again, I'll draw the purine ring. Okay, better if I can copy the same thing. Else it will take me too much time to draw this. Yes. Okay. Right. So I'll convert this back to, yeah. So here you have the purine ring of yours. I'll erase the double bonds for now because, okay. So here you have the raw structure. Now the name says 2 amino 6 oxypurine, right? 2 amino. So I'll put the amino group over here, over the second carbon. I'll put the amino group, that is the NH2 group, and over the 6th carbon I'll put the oxy group. Okay, here you have the oxy group. Now I will have to satisfy the valency of the carbon atoms now. So here I will have to put a double bond, here I will have to put a double bond. Nitrogen mein, here you will have the hydrogen. Okay, and then here. Okay, so now the valency is all satisfied. You can look at the structure. Yes, the valency is satisfied. So this is the basic structure for guanine. So I have taught you how you can remember the structure for adenine and guanine. Now let's move on with the structure of cytosine. So cytosine. Then I will teach you about the structure of uracil, and then I'll teach you about the structure of thymine. Let me draw the structure for pyrimidine first, and I'll not put the double bonds. I'll keep the double bonds for later. Okay. Now, so since we know that cytosine is also known as 2-oxy-4-amino pyrimidine. Okay, so I'll write these names as well. It is 2-oxy-4-amino pyrimidine. Uracil, it is known as 2,4-dioxypyrimidine. And thymine is known as 5-methyluracil. Okay, sure. Let's start with the structures now. 1, 2, 3, 4, 5, 6. Okay, so this is how the numbering would be done. Now 2-oxy, so 2 here, 2-oxy, so I'll put a double bond here. 4-amino, so 4-amino, NH2 group here. Okay, NH2 group here, and then I'll have to justify the valency. So here you will have a bond, here you will have a bond, right? If you can look at the second carbon, the valency of the carbon is all satisfied, so I'll have to put a hydrogen over here. Okay, so this is how the structure will look like. I'll shift this down. Yes. Okay, this is how the structure should look like. Then comes the uracil. So 2,4-dioxypyrimidine again will make the pyrimidine ring first. Okay, then let's do the numbering now. 1, 2, 3, 4, 5, 6. Okay, so 2,4-dioxy. Second position पे oxygen, here oxygen. Okay, so you can see the valency of carbon is satisfied at carbon at position number 2 and 4. So here I will have to put a double bond. Here I'll have to put a double bond. Now nitrogen is left, so one hydrogen here, one hydrogen here. So this is going to be the structure for uracil. Again, from the uracil, it is easy. It is very easy to make thymine. Thymine is 5-methyl uracil. Okay, so what will I do? I'll copy this structure for you. Uracil exact name. I'll copy this and I'll paste it over here. Okay, now I know it is 5-methyl uracil. So over the fifth carbon, I'll just have to add the methyl. I'll carry it methylation. Okay, so here you have the structure for cytosine, uracil, and thymine. If you want, you can also remember these structures from here, right? So I have added these one all as well. So adenine, guanine, thymine, cytosine, and uracil. This is how you can memorize. Okay, I have taught you how you can draw the structure for these compounds. Now let's talk about what is a nucleoside. What is a nucleoside? So nucleoside, sugar plus nitrogen base is called as nucleoside. Let's draw this. Okay, so this is the pentose sugar. As I said, beta form it should be. So here you should have the OH group on the upper side, right? We are doing, let's say, ribose sugar. So here I will have OH. Here I will have OH. Here I'll have CH2OH. Okay, so this is the structure for the ribose sugar, right? So with the first carbon, okay, here you will have the nitrogen base attached. Okay, here you will have the nitrogen base. So this is a nucleoside. This is the structure for a nucleoside. Sugar plus nitrogen base. Correct? Sugar plus nitrogenous base is known as nucleoside. Okay, so a nucleoside, again there will be some naming for it. So I'll write the names. So the adenine, guanine, these are the nitrogen bases. Okay, adenine, guanine, then you have cytosine, thymine, and then you have uracil. So in the case of purine, if I have written adenine, so this is the name of the nitrogen base. If I have to write the name of the corresponding nucleoside, for the purines you need to add cosine at the ending of the term. Okay, so adenine will become adenosine, adenosine. Guanine will become guanosine. Okay, for the case of, in the case of pyrimidines, you need to add idine in the ending. Cytidine, thymidine, uridine. Okay, cytidine, thymidine, and uridine. So when I say adenine, you should get in your mind that I'm talking about the nitrogen base. But when I say adenosine, you should know that I'm talking about the nucleoside. What is the nucleoside? You will have the nitrogen base, and to which the sugar group will be attached, right? So you will have the sugar attached to it. Now let's talk about the nucleotides. What are the nucleotides? So nucleotides, we have already I have already taught you about this, that when you add the sugar plus nitrogen base plus phosphoric acid or phosphate group, right? It is called as nucleoside. So sugar plus nitrogen base, we already know that it is a nucleoside. So nucleoside to which the phosphate group has been added. Phosphate group has been ester, like the ester bond will be formed. Okay, so that will be the nucleoside. Again, if I have to draw the structure, I'm making a ribose sugar for you. Here you have the nitrogen base. Okay, OH, OH, CH2, OH, right? So with the hydroxyl group, let's draw the phosphoric acid. Okay, so I can establish a bond like this, correct? I can establish a bond like this. So I'll erase this, and a bond will get established. Okay, so this is the structure for the nucleotide. So nucleotide, nucleotide. Yes, so we can write the corresponding names for each and every single nitrogen base, right? So nucleotide, if you can see, you have the nitrogen base, then you have the sugar. So nucleoside plus phosphate. You have one phosphate group, so monophosphate. So nucleoside monophosphate, right? So nucleotides are what? They are nucleoside monophosphates. Okay, so you have nucleoside monophosphates. So you can have adenosine monophosphate. You can have guanosine monophosphate. You can have cytidine monophosphate. You can have thymidine monophosphate. And finally, you have uridine monophosphate. Okay, now these are the names you have. So they have another name as well. So adenosine monophosphate is also known as adenylic acid. Please make a note of this. It is also known as adenylic acid. Okay, guanosine, guanalic acid. Guanalic acid. Cytidine, cytidylic acid. Thymidine, thymidyllic acid. Uridine, uridylic acid. That's it. Okay. So adenosine monophosphate, adenylic acid, guanosine monophosphate, guanylylic acid, cytidine monophosphate, cytidylic acid, thymidine monophosphate, thymidylic acid, uridine monophosphate, uridylic acid. These are the common names for these compounds. Okay, and I have also taught you how you can draw the corresponding structures for these. So very easy for you. Now let's move on to the next one, that is DNA. What is DNA? So DNA is helically twisted polydeoxyribonucleotide. Okay, what is it? What is DNA? It is helically twisted. Okay, it has two chains, so I can mention this double chain polydeoxyribonucleotide. Okay, so DNA serves as the genetic material for all living organisms except riboviruses. So it is the genetic material for all living organisms except riboviruses. Why? Because riboviruses will have RNA as their genetic material. So I'll underline this. Okay. So DNA, it is the largest macromolecule. It is the largest macromolecule among all the biomacromolecules. It is the largest macromolecule with the dimensions as its diameter will be the diameter, that is the distance between the two strands of the DNA. Diameter is somewhere around 2 nanometer or 20 angstrom. 20 angstrom. And the length will be in millimeters. Length will be in millimeters. Okay, length will be in millimeters, right? So this much information you need to know. I can also give you about the count of the base pair. Let's say in human haploid content. Okay, I'll write this thing. In human, the haploid content is 3.3 into 10 to the power 9 base pairs. 3.3 into 10 to the power 9 base pairs. That is 3 billion, approx 3 billion base pairs we have. One more, right? E. coli, Escherichia coli, that is 4.7 million. 4.7 million base pairs are there in the case of Escherichia coli, correct? So now DNA, you have two strands. The two strands are anti-parallel to one another, and the whole duplex is helically twisted. The whole duplex is helically twisted around an axis. Okay, I hope everyone's clear. Like, I hope you would, if you would have visited any of the fancy cafes or restaurants, you would have seen this staircase which is twisted, right? So the two, right, the staircase, this you can visualize that is the structure of the DNA, right? The helically twisted staircase. So you have two strands. They are not twisted over one another. Instead, the whole duplex, right? The two strands, they remain antiparallel and they are twisted like this. Okay. So helically twisted double chain polydeoxyribonucleotide is the DNA, that is a deoxyribonucleic acid. I hope this is clear to all of you. So now you should know that how the DNA looks like. So I'll come to this particular diagram for you. That this particular diagram, what does it show? So first of all, we have the sugar phosphate backbone. Sugar phosphate backbone. So sugar phosphate backbone means we have the phosphodiester linkage. What we have? The phosphodiester linkage. Okay. And the nucleotides, they are perpendicular to the sugar phosphate backbone, and the nucleotides, they pair together with the help of hydrogen bonds. Okay, so I'll teach you how the sugar phosphate backbone gets formed, why the two strands are anti-parallel to one another. Let's move on to that point first. So let's talk about the phosphodiester bond. Okay, so again chemistry. Let's draw the structures first. Now I will draw the structure for two nucleotides. Okay, I want to establish the phosphodiester bond between two nucleotides. So I'll draw the structure for two nucleotides. Okay, so if you can see here, we have two nucleosides. I have drawn two nucleosides right now. Okay, so the first one is the adenosine, the next one, the adjacent one, is the guanosine. Okay, so now first of all, these will have the phosphate groups, right? These will have the phosphate group. Okay, so I will attach the phosphate group over here. Okay, so the phosphate group here, and also you will have one phosphate group here. So instead, I am drawing it in this. Okay, okay, correct. So now if you can see, I'll do the numbering first. So adenine and guanine, there also you will have the numbering, and here in the ribose also, the pentose sugar, here you will also have the numbering, right? So to distinguish which compound I am talking about, for the ribose, for the sugar, we do the numbering with a prime, like with a dash to it, right? I'll show you. Like here, so it will be called as 1 dash, 2 dash, 3 dash, 4 dash, and here you have the 5 dash, or the 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. This is how you can pronounce it. Okay, so here I can see that over the fifth carbon, right, at the 5 dash position, I have the phosphate group here also and here also. 1, 2, 3 dash, 4 dash, 5 dash, right? So over the 5 dash carbon, you have the phosphate group attached. So here this is the ester bond, right? This is the ester bond, correct? Now if you can see the third carbon as the 3 dash carbon of the sugar, right over here, right here, one more ester bond can be established. Okay, so I can draw it like this manner, OH. This is how the bond can be established. Correct? Correct? Okay. So I'll erase this. I'll erase this and I'll put a bond. Okay. So this is an ester bond. Here you have the ester bond. Here also you have the ester bond with the 3 dash group, right? So this is the phosphodiester bond. Correct? I hope this is clear how the phosphodiester bond gets established. If you can see, this is a dinucleotide. You have two nucleotides to dinucleotides. Now, if you can see here, you have one 3 dash. Okay, as I taught you that the phosphodiester bond gets established with the 3 dash carbon, sorry, 3 dash to the phosphodiester bond gets established with the hydroxyl group attached to the 3 dash carbon of the sugar, right? And with the 5 dash carbon, you have the phosphate group. So this shows the polarity of the like of the chain, right? So 3 dash and represents a free hydroxyl group, free hydroxyl group, right? Whereas the 5 dash end represents the free phosphate group, free phosphate group. This is what you need to know, correct? Now I can come to the structure of the DNA. So you can see here we have the sugar, then attached to which is the nucleotide adenine. Okay, and then we have the phosphate group. So if you can see, this phosphate group is free, so this is the 5 dash end. This is the 5 dash end. Let's look at the structure in detail. Over here, if you can see, this is the sugar. Now at this sugar, you have the 3 dash, at the 3 dash carbon you have the free OH group, so this is the 3 dash end. Okay, and here we have the established phosphate group. Okay, so this is the phosphate bond. So this is what you need to know. Okay, here we have the phosphodiester bond. This is the structure, the molecular structure for the DNA. Now you need to know that the two chains are held together by the hydrogen bonds. Okay, so between A and T, adenine and thymine, we have two hydrogen bonds. Between G and C, we have 3 hydrogen bonds. So here I will have 3 hydrogen bonds. I will have 3 hydrogen bonds. Okay, so this is the molecular structure for the DNA. What we need to know first, about the polarity. The DNA has polarity. Okay, you have one strand with the polarity 3 dash to 5 dash, and the next strand is with the polarity 5 dash to 3 dash. Okay, that means we have two strands, and the two strands are anti-parallel in nature. The strands are anti-parallel in nature. The two strands are anti-parallel in nature. Okay, so I'll tell you about this. Like, let's say I have written a sequence A, G, C, T. Okay, A, G, C, T. So this is the 3 dash and this is the 5 dash. And if I'll write, okay, so T pairs with A. So here I will have A, C pairs with G, G pairs with C, A pairs with T, T. Okay, so if you see the two strands, they are anti-parallel and they are complementary with one another. So next, the two strands are anti-parallel, the polarity I have taught you, and then the complementary. The two strands are complementary. Okay, the two strands are complementary with one another. Okay, now one logic by, if you can see, as I told you, that adenine pairs with thymine, guanine pairs with cytosine. Okay, so what happens here, that a purine is always pairing with a pyrimidine. Why a purine cannot pair together, or why a pyrimidine cannot pair together? The answer is very simple. When a purine, like 2 rings, pyrimidine 1 ring, total 3 rings. So you get a very symmetric structure. But let's say if the purines are pairing, pyrimidine rings are pairing, so what will happen? 4 rings, 2 rings, 4 rings, 2 rings, 4 rings, 2 rings. So that will become a very unstable structure, right? Let's say you are making a bundle of books. So in one manner, what you can do, you can place all the similar books of similar sizes, right? You can stack them together, then the smaller one, then more smaller one, right? The smallest one. That's how it will start looking like a pyramid, like quite stable. But if I put, like, let's say 3 big books, then 2 or 3 smaller books, then bigger books, then smaller books, bigger books, smaller books, so that structure will not be that sturdy, right? It will be highly unstable, right? So that's why a purine always pairs with a pyrimidine. This is what you need to understand. Now, some points. As I said, the two strands are anti-parallel. There is hydrogen bonding between the two nitrogen bases. You have 2 hydrogen bonds between adenine and thymine. You have 3 hydrogen bonds between guanine and cytosine. Okay, the duplex itself is helically twisted. These are the most important points that you need to know. Now, some numbering. This is the structure of the DNA. Now, let's talk about some numbers. So the distance between the two strands is about 2 nanometer. Converting it to angstrom, so it is 20 angstrom. First point. Now this will be helically twisted. So upon twisting, what happens? You get like one complete 360 turn. One complete 360 turn will have 10 nucleotides. Okay, so one complete 360 turns will have 10 base pairs. Okay, so the pitch is about 34 angstrom. The pitch is about 34 angstrom, correct? So 34 is the complete pitch, and in the whole pitch you are getting 10 nucleotides. So matlab distance between two base pairs, right? Distance between two base pair will be equal to 34 upon 10, that is 3.4 angstrom, right? So I have to write it in nanometer. I can write 3.4 nanometer. Here it will become 0.34 nanometer. Okay. Now, when this helical twisting happens, you get grooves in the DNA. So you can see here the bigger groove. This is the major groove. And here, if you can see here, this is a minor groove. Okay, so the diameter for minor groove is let's say 12 angstrom, and for the major groove it is 22 angstrom. Okay, it is 22 angstrom for the major groove. these group, what are the functions of the minor groups and major groups? So these are the sites where, like, the, let's say, the transcription factors, okay, or whatever the important enzymes, they come and they bind to the DNA. So if anything has to interact with the DNA, it will come and it will bind to the minor groups or the major groups. This is a function. Okay, so that's all from my side for today's class, guys. This is all about the DNA. Now if you have any doubts, you can ask me, you can mention them in the comment section. From the very next class, I'll continue with the structure of the RNA, lipids and enzyme classification. So hopefully the next class is going to be the last second class for this particular chapter. Okay, so I need 2 more classes and we'll finish this chapter. That's all from my side. Thank you very much. See you in the next class.